What you're seeing on screen is a single solitary neuron searching for connections. In this time-lapse, we can watch the neuron's axon and dendritic branches grow increasingly complex, reaching out for contact points with other neurons. This process is what triggered every memory you have, every smell you've ever experienced, every feeling of anger, love, compassion, or shame, everything you think you are, everything you are but don't even know you are, everything you love or hate about yourself. It all begins with this tiny neuron reaching out for something to connect to. In the winter of 1906, two scientists shared the Nobel Prize in Physiology or Medicine and used their acceptance speeches to publicly contradict each other. Camillo Golgi and Santiago Ramon y Cajal had spent decades studying the same tissue, using the same staining technique that Golgi himself had invented, and arrived at opposite conclusions about one of the most fundamental questions in neuroscience at the time: whether the brain was a continuous web of interconnected matter, or whether it was made up of billions of individual cells, each separate from the next. Golgi believed it was a web. Santiago believed it was made of individual cells. And standing in Stockholm, receiving the same prize, each man argued that the other was wrong. Santiago turned out to be right. The brain is built from individual units, neurons, and what happens between them, across the tiny gaps that separate one from the next, helps produce everything you think, feel, remember, and decide. Every emotion you've ever had, every habit you've built, every memory that surfaces unexpectedly at 2 a.m. All of it depends on neurons firing, connecting, strengthening. What happens in those gaps and how neurons integrate signals to decide when to fire and when not to turns out to explain far more about daily human experience than most people expect from a biology lesson. A neuron is not a wire. That's the first misconception worth clearing up. Thinking of neurons as wires carrying electricity from point A to point B makes the brain sound like a circuit board. The reality is considerably stranger than that. A neuron is a cell. It has a body, a long tail called an axon, and a branching set of receivers called dendrites. When a neuron fires, it sends an electrical signal down its axon. But that signal doesn't jump directly to the next neuron. It reaches the end of the axon and triggers the release of chemical messengers called neurotransmitters, which cross a tiny gap called a synapse and act on the dendrites of the next cell. The receiving neuron then has to respond. That response isn't automatic. A neuron receives signals from thousands of other neurons simultaneously, some telling it to fire, some telling it not to. Whether it fires depends on the balance of those signals at that moment. It's less like a switch and more like a vote, with thousands of inputs arriving at once and the neuron responding to the net result. This is happening across roughly 86 billion neurons, each connected to thousands of others, producing on the order of 100 trillion synaptic connections in a single human brain. The number is almost impossible to hold in your head meaningfully. But what matters about it isn't the scale. It's what that scale makes possible. Because the brain isn't storing information in individual neurons alone, it's storing it in patterns, in the specific configuration of connections between neurons, and in how strong or weak those connections are. A memory isn't located in one cell. It's distributed across a network, encoded in the relationships between many cells that learn to fire together under specific circumstances. This is why a smell can pull up a memory from 20 years ago with a vividness that a deliberate effort to remember the same moment often can't match. The neurons associated with that smell and that memory were activated together and can later reactivate related activity patterns. The pattern storage system that neurons use has a property that took researchers a long time to fully appreciate. It's not fixed. Donald Hebb, a Canadian psychologist, proposed in 1949 what became one of the most cited ideas in neuroscience: that neurons that fire together wire together. When two neurons activate at the same time repeatedly, the connection between them can strengthen. The synapse becomes more efficient, requiring less signal to produce a response. Over time, activating one neuron makes it easier to activate the other, until the association can become almost automatic. This is one important part of how learning works at the cellular level. Skills, memories, behavioral patterns that stop requiring conscious effort. All of it can involve synaptic connections becoming stronger through repeated co-activation. But the same mechanism that makes learning possible also makes certain things difficult to undo. Anxiety is a useful example. When the brain repeatedly pairs a situation with a threat response, say social interaction, with a feeling of danger, the neurons involved in that pairing can strengthen their connection. Over time, the situation alone may be enough to trigger the response, even without any actual threat present. The amygdala can activate faster than the prefrontal cortex has a chance to fully evaluate the situation, which means that by the time the rational part of the brain starts assessing what is happening, the emotional response may already be underway. That's why anxiety often feels illogical from the inside. The emotional response isn't waiting for a reasoned assessment, it's firing along a pathway that was built before the reasoning even started. Memory follows the same logic, but in a direction most people find surprising. The brain doesn't store memories like photographs. Every time you recall a memory, you're reconstructing it from the pattern of neurons that were active when it formed. And that reconstruction process isn't neutral. It's shaped by whatever the brain is going through at the moment it's trying to remember. Elizabeth Loftus at the University of California, Irvine, spent decades documenting how memories can shift each time they're accessed, influenced by subsequent experiences, by suggestion, and by the emotional state at the moment of retrieval. What feels like remembering something is often closer to rebuilding it, with whatever materials are currently available. The neurons involved in the original experience and the neurons active during retrieval interact, and the memory that gets stored afterward can be updated in subtle ways. And that has implications that go well beyond memory research. The stories people carry about themselves, about who they are, what they're capable of, what certain experiences meant. Those aren't static records sitting somewhere in the brain waiting to be retrieved. They're active constructions, reshaped every time the relevant neural patterns get reactivated. The version of neuroscience that gets taught in school, it's accurate enough to pass a test, but it leaves out the parts that are actually strange. One of them is pruning. The human brain is born with far more synapses than it will ever keep. In the first years of life, the brain is building connections at a rate that will never be matched again. And then, through childhood and adolescence, it begins systematically eliminating the ones that aren't being used as much, a process called synaptic pruning that has nothing to do with deterioration. It's the brain becoming more efficient, cutting what experience is deemed unnecessary and strengthening what is proven useful. The brain you have as an adult was shaped by what you were exposed to, what you practiced, and what you paid attention to repeatedly enough that the relevant connections survived. Michael Merzenich at the University of California, San Francisco, spent decades mapping how the brain's sensory regions reorganize in response to experience. In classic studies, when sensory input was altered or lost, the brain region dedicated to processing that input didn't go quiet. Nearby regions gradually took over parts of its territory. The brain redistributed its resources based on what was actually coming in. And that reorganization isn't limited to sensory regions or dramatic physical changes. It happens in response to repeated thought patterns, habitual behaviors, and sustained attention. The structure of the brain reflects the history of how it's been used. Neuroscientists call this neuroplasticity, a term that's been overused enough to lose some of its weight, but the underlying reality is more radical than the word usually suggests. Something else that rarely gets mentioned is how much of the brain's activity has nothing to do with conscious experience. At any given moment, the vast majority of what your neurons are doing is happening beneath the level of awareness, regulating systems you never think about, processing sensory information before it reaches consciousness, generating predictions about what's likely to happen next based on everything that's happened before. The conscious experience of making a decision is a small fraction of what the brain is actually doing, and it arrives downstream of processes that were already underway before any awareness of them surfaces. That reorganization the brain does in response to experience doesn't just happen during childhood or in response to dramatic events. It happens in response to what you do every day. repeatedly, without much thought. And that's where the neuroscience starts to feel personal. Anne Graybiel at MIT spent years studying how the basal ganglia helps encode repeated behaviors. What her research showed is that when you perform the same sequence of actions in the same context repeatedly, the brain can gradually compress that sequence into a more automated chunk. The individual decisions that made up the behavior get folded into something that runs with far less conscious involvement than it required at the start. That's what a habit is, at the neural level. Not a character trait or a matter of discipline, but a compressed sequence of neural activity that the brain has learned to run efficiently. And that process has no moral filter. The brain automates what gets repeated, regardless of whether the behavior is useful or destructive. The neural pathway for reaching for your phone when you're bored gets strengthened by the same learning machinery as the pathway for sitting down to work. Both are patterns that have been reinforced enough times that the brain starts treating them as defaults. Most people approach unwanted behaviors as if the problem is motivation, as if wanting to stop badly enough would be sufficient. But the pathway is already built. The neurons involved have already been connected through repetition, and wanting to stop doesn't automatically dismantle the circuit. That's why willpower alone so rarely works. It's a conscious effort competing against an automated system. And automated systems tend to win that fight unless the environment changes too. What actually weakens a neural pathway is disuse. Connections that stop being activated tend to become less efficient over time. And that means the most effective way to change a behavior isn't to fight the existing pathway directly, but to build a competing one through repetition, in a consistent enough context that the brain starts treating the new pattern as the default instead. Charles Duhigg, drawing on Graybiel's research, described this as replacing the routine while keeping the cue and the reward intact. Because the cue has already been wired to trigger something, and redirecting where it leads is easier than trying to sever the wiring entirely. Santiago spent the last decades of his life drawing neurons by hand, thousands of them, rendered in extraordinary detail with pen and ink, trying to capture structures so small they were invisible to the naked eye. He was working before electron microscopes existed, before anyone could confirm what the gaps between neurons actually looked like up close. His drawings were based on what he could see through a light microscope and what he could infer from the patterns. And he got most of it right. What he couldn't fully appreciate at the time was what those gaps would eventually turn out to mean. Because it's not the neurons themselves that store who you are. It's the connections between them. The specific configuration of which cells talk to which other cells, how efficiently, and under what circumstances. That configuration keeps shifting across a lifetime, shaped by repetition, by attention, and by everything that got left alone long enough to stop mattering to the circuits involved. The brain Santiago was drawing is the same brain running every conversation you've had today, every decision, every moment where something felt familiar or didn't. And it's not the same brain it was 10 years ago. Not because the cells are different, but because the pattern of connections between them has been quietly reorganizing itself the entire time, based on everything that happened in between.